Thank you for joining me. Today we will go through Biology Paper 1, and this is June 2021 paper, and this is for the AQA GCSE Combined Science Trilogy course. Okay, so let's make a start. So plants absorb light for photosynthesis. Which is the equation for photosynthesis? Tick one box. So what you need to do here is have a look at the reactants and products. So we know that during photosynthesis glucose is produced, so we need to find glucose after the arrow. So we know it can't be this one, so it's either this one or that. We also know that oxygen gas is produced during photosynthesis, and I can only see oxygen gas here, so this is the correct option. A student investigated the effect of light intensity on the rate of photosynthesis. Figure 1 shows the apparatus. So this is your photosynthesis required practical. So you've got your pondweed in a test tube, you've got bubbles of gas, you've got your lamp, and you've got your ruler here, which shows distance from lamp in centimeters. This is the method used. Set up the apparatus as shown in figure 1. Place a pondweed 10 centimeter away from the lamp. Switch on the lamp. Record the number of bubbles of gas produced in 5 minutes. Repeat steps 2 to 4 with the pondweed at different distances from the lamp. What was the independent variable? This is the variable I will change in this investigation. So if you go back to step 5, it says repeat steps 2 to 4 with the pondweed at different distances. So that means I am changing the distance of the pondweed from the lamp. The lamp gets warm when it is on. This causes the temperature of the water to increase. Explain how an increase in temperature would affect the results of this investigation. So it's an explain question, so this question involves science. So if you increase the temperature, the bubbles would be produced faster, bubbles of oxygen. So bubbles would be produced. It's 2 marks, so all I want you to do is just write down 2 bullet points. Keep it nice and simple. So why are we producing more bubbles? Why are we producing bubbles at a faster rate? It's because at higher temperatures, enzymes are working faster. Suggest one way the investigation could be improved so the temperature of the water does not increase. So if you go back and look at the setup, when that lamp is on, that water surrounding the podweed is going to get warmer. So how do we stop that from happening? You can use an LED lamp, which is the easiest option. Okay, or if you think back to when you studied this required practical in your lesson, you should have learnt that what you can do is, I know I don't have much space here, but you can use a large beaker, fill it up with cold water. So a large glass beaker, fill it up with cold water and place that between your lamp and your pondweed. And the heat from the lamp would be absorbed by this cold water, which means that would prevent the water surrounding the pondweed from getting warmer. But how would you put that as a bullet point? I know it's 1 mark, so you can either use an LED lamp, or you can say place a beaker of cold water between the lamp and the test tube. And the tube. That's it. So either this or that. Question 1.5 suggests 2 improvements to the investigation so the results would be more valid. Do not refer to controlling the temperature of the water. So here you can say, so we're counting the bubbles. Why don't we measure the volume of gas? So measure volume of gas produced. Or you can even say use a gas syringe to collect the gas. That's one marking point. You can say use the same lamp, or allow the pondweed time to acclimatize. The pondweed won't start producing oxygen bubbles straight away. You need to give it time to adjust to the new conditions. So you would say allow the pondweed time to acclimatize. Or you can just say use the same lamp. Right, next question. Calculate the rate. So this is the data. Calculate the rate of photosynthesis when the pondweed was 40 centimeter from the lamp. Give the rate of photosynthesis as the number of bubbles of gas produced per minute. So let's have a look. So we're looking at 40 centimeters and I've got 16 bubbles produced in 5 minutes. I want bubbles produced per minute. So I do 16 divided by 5. Just bear with me while I get a calculator. That gives me 3.2, but we're not going to produce 3.2 bubbles, so round it down to 3 bubbles. If you just left your answer as 3.2, you should still get 1 mark. Give one conclusion that can be made from table 1. So we can see that as the distance increases, that means as the distance is increasing, light intensity is going down because I'm moving the lamp away, and you've got the number of bubbles, you can see that's decreasing. So you can say as light intensity decreases, the rate of photosynthesis decreases. So as light intensity decreases, the rate of photosynthesis decreases, or you can say as the distance from the lamp increases, the number of bubbles produced decreases. That is still a valid answer. That should get you 1 mark. Now plot the data from table 1 on figure 2 and then draw a line of best fit. So let's start plotting the results. So 10, 120, 20 is 56. So each square is worth 2 digits. 30 is 31. 40 is 16, so that's 10, 16, and then 50 is 10. So always go back and check, right, have I plotted all the data? Yes, I have. Draw a line of best fit. Okay, so if you have a look, it's a curve. So no use of a ruler, freehand, smooth curve. Okay, so that's slightly out, but it's possible to get that onto a curve. Okay, so freehand Okay, don't use a ruler, no dot to dot. In science, a line of best fit can be a straight line or it can be a curve. So I've done my line of best fit. Always tick off what you've done. Right, predict the number of bubbles that would be produced in 5 minutes if the pondweed was 60 centimeter from the lamp. Well, I don't have any data points for 60 centimeters, so what I do is I can predict using my graph. So I'm just going to extend my line, and I know it's going down. Okay, extend the line, then use a ruler, go up to so start from 60, go up, draw a line vertically up until it touches the line, draw a line going across, read that value off. Okay, and from my graph, it's so you've got 4, so 4 bubbles produced in a minute. Okay. Next question. And again, this answer here will vary. So if you do look at the mark scheme, if you don't see your answer, it's not the end of the world, because it depends on your line of best fit and how you've brought the line down. Okay, and as long as you've read that value off correctly, it should be fine. Okay. Describe, so question 2. So this is your food test required practicum. Describe how to test a sample of food for protein, starch and sugar. Give the colours that would be seen if the food sample contained protein, starch and sugar. Right, so the best way to answer this question is have subheadings. You are allowed to have subheadings. So let's look at how we test for protein. You don't need to give lots of details about how you prepare the food sample. Keep it nice and simple, and all you can say is grind up the food. You don't need to mention pestle and mortar. Right, for burette, I would add burette solution. You don't need to add the volume. And then the question doesn't say mention the original color. It's what would be seen. So what you should see is if it turns purple, protein is present. Okay, so if it turns purple, protein is present. That's it, done for protein. Let's have a look at starch. So for starch, I would add iodine solution. For this one, you don't need to grind up the food. You can use the food sample as it is. So add iodine solution. And if it turns black, iodine is present. Sorry, starch is present. Okay, and then my last one is sugar. Sugar is just slightly complicated. For sugar, you have to grind up your food. So grind up the food. And you can say add Benedict's solution. And there's an extra step here, which is about heat the mixture. If you want to mention how you would heat that mixture, you can heat the mixture in a water bath. And if it turns, let's just say brick red, sugar is present. Okay, and there's different colours. You can have brick red, you can have orange, you can have brown, you can have yellow, you can have green. The different colours indicate the amount of, or the concentration of sugar in that particular sample of food. Okay, but you don't need to mention all the colours, you can just mention one colour. Go with one colour that you will remember. I tend to go for brick red. If you have all of that, that should get you 6 out of 6, because this method will lead to the production of a valid outcome. I've got all my steps, and all my steps are logically sequenced. Okay, and to get your 5 or 6 marks, you must make reference to all 3 tests. Okay, question 3, fermentation. So this links to the very last topic of biology paper 1, which is bioenergetics, respiration. So fermentation is an exothermic reaction. What does exothermic mean? It's when energy is released to the surroundings. Or just keep it simple. It's a reaction that releases energy. Okay? A student investigated the effect of temperature on fermentation in yeast. Figure 3 shows the apparatus. You've got a gas syringe, you've got a flask, I've got a layer of oil on top of my yeast. And this is the method. Mix yeast with sugar, put a layer of oil, put the flask in a water bath, leave for 20 minutes, add a gas syringe, collect gas every 5 minutes for 30 minutes. After 30 minutes, move the flask to a water bath at 35 degrees Celsius and continue to record the volume of gas collected every 5 minutes. Suggest why a layer of oil was needed on the surface of the mixture. Now here, look at your equation. It's glucose, breaks down into ethanol and carbon dioxide. No oxygen here. Okay, because if you want to produce ethanol and you want to produce carbon dioxide, there should be no oxygen. Okay, so fermentation is basically anaerobic respiration. So that layer of oil is essential because it keeps oxygen out. And that's your answer. Suggest why the mixture was left for 20 minutes before the gas syringe was attached. So this bit here. And this is what I mentioned earlier when I was talking about the photosynthesis required practical. It's all about acclimatizing, allowing things to adjust to their surroundings. So here, the answer would be to allow the mixture to reach 2 degrees Celsius. So to allow the mixture to reach 2 degrees Celsius or to reach the temperature. Okay? And then you've got your results here. Steps 1 to 4 of the method are repeated at 35. And again, volume of gas collected every 5 minutes for 45 minutes. Table 2 shows the results for both flasks. And then table 3 shows the results of both flasks when they were left at 35 degrees Celsius. Okay? So take your time, have a look at that data. Explain the results from 0 minutes to 45 minutes for the flask that was at 2 degrees Celsius and was then moved to 35. So we're explaining all these zeros and then the 2, the 7 and the 22. So you start off by saying, it says 3 marks, 3 bullet points. 2 degrees Celsius is too cold for enzymes. Do not say denature, because enzymes do not denature at 2 degrees Celsius. They just become inactive. So if the enzymes are inactive, no CO2 is produced. That's 2 bullet points, so I get 2 marks. Because I've got 0 centimeter cubed, that's telling me no carbon dioxide is produced, or you can just say no gas is produced. And then let's explain the 35, what's happening at 35 minutes, 40 minutes, 45 minutes. So at 35 degrees Celsius, enzymes become active. So CO2 or gas is produced. That's it. The question comes across really complicated, but trust me, it isn't. Next question, explain the results from 0 minutes to 45 minutes for the flask that was kept at 35. So now let's repeat, okay, for the second flask. So you can see it's increasing, so the amount of gas produced increases. But then if you have a look at 35, 40, 45, it tends to kind of stay the same. So why is that happening? Make sure you use table 2 and table 3. So here we'd say that the temperature, because we've got figures here, there's gas being produced, the temperature is suitable for the enzymes. And carbon dioxide is produced, so CO2 is produced, or you can just say gas is produced. And then let's have a look. So you can see roughly around about here, fermentation slows down. So you can say eventually fermentation slows down. That's 3 bullet points, 3 marks. And now why has it slowed down? If you look at your equation, let's take you back. So remember, this is the gas that we're collecting. This gas is produced when glucose is broken down. If I'm not producing carbon dioxide, that probably means that I've run out of glucose. Okay, so your 4th bullet point should say glucose began to run out, or the sugar began to run out. Okay, that's it, and that should get you 4 marks. Again, they've given you lots of lines to write down the answer. Trust me, you don't need to fill up every single line. And you know, I suggest, in order to stop the waffling, you just write your answer as bullet points. Okay, next question, question 4. So this question is looking at infection and response. So draw one line from each disease to the pathogen that causes the disease. So gonorrhea is caused by bacteria, malaria protist, measles is virus. So there's 7 diseases that you need to be familiar with in the AQA spec. So gonorrhea is an example of a disease caused by bacterium. The other one you need to know is salmonella. Fungus, you need to know rose black spot, which is a fungal infection that occurs in plants only. Protist is malaria. Virus, you need to know HIV, TMV, tobacco mosaic virus, that's plants. And you need to know measles is already there, so 1, 2, 3, 4, 5, 6, 7. Okay, so there's 7 diseases on the spec. So make sure you look at all of these and make sure for each you know prevention. How would we prevent this disease from occurring? All 7, and you need to know mode of transmission. So method of transmission. How is it spread from person to person? Malaria is transmitted by mosquitoes. Male mosquitoes can be sterilized, so they are infertile. The spread of malaria is reduced by releasing sterile mosquitoes into the environment. Explain how releasing sterile mosquitoes reduces the spread of malaria. So, very clear, if we're sterilizing, that means you are reducing the breeding rate. So reduces breeding or reduces reproduction. That means I will have fewer mosquitoes biting people. And that's my 2 points made. Okay. Pathogens also cause disease in plants. Shows a rose black spot fungal spore and a tobacco mosaic virus. Yeah. Name the piece of equipment used to view the virus. Now, virus, look at that. Tiny. Okay. That means the microscope that is used is an electron microscope. There's no way you can view a virus under a light microscope. That's electron microscope. Next question. How many times longer is the fungal spore than the virus? So what you need to do here is convert. So both units need to be the same. Okay, so let's go for 16 micrometers. Let's convert that to meters. So the best way to do that is, so in 1 micrometer, actually hold on, let's just do it the other way. So in 1 millimeter, you have 1,000 micrometers. Okay. 1 meter is 1,000 millimeters. Yeah. So if 1 millimeter, is 1,000 micrometers, that means 1,000 millimeters would be 100,000 micrometers. Okay. So to get from meters to millimeters, I multiply by 1,000. To get from millimeters to micrometers, I multiply by 1,000. Okay, so you need to know these conversions. So I'm working with 16 micrometers. I want 16 micrometers to be converted into meters. Okay, because if you have a look here, tobacco mosaic virus, the unit is meters, that's micrometers. I can't work out how many times longer if my units are different. So I prefer to convert micrometers to meters, or you can do meters to micrometers. It's your choice, okay? But I'm going to go from micrometers to meters. If I've got 16 micrometers, let's take that to millimeters. So if I'm going in reverse, that's going to be divided by 1,000. So that would be 0.016. I need to divide that by 1,000 again to get to meters. So on your calculator, if you divide that by 1,000 again, that should give you 0.00016. Okay, so that 0.016 divided by 1,000. So I've converted it into meters. Now the next step is, so I would go, if you want to write that in standard form, that's basically 1.6, because my calculator doesn't give the answer in standard form, but yours probably does. 10 to the power of minus 5, 1, 2, 3, 4, 5, yep. And I'm going to divide that by 2.5 times 10 to the power of minus 7. So if you put that into your calculator, you should get 64. So the number of times longer, so is 64. So the fungal spore is 64 times longer than the virus. Okay, and that's 3 marks. Explain why the plants infected with tobacco mosaic virus grow slowly. So remember, with tobacco mosaic virus, the leaves are discolored. Okay, so that's something you need to know. So you need to know the symptoms for each as well. So you can add that. So the leaves are discolored. And the reason why there's discoloration there, it's because there's lack of chlorophyll. Or you could have written that, less chlorophyll in the leaves. If there's less chlorophyll in the leaves, there's less photosynthesis or reduced photosynthesis. If I have reduced photosynthesis, I will have less glucose produced. So less proteins, which is why there's lack of growth, which is why plants infected with tobacco mosaic virus grow slowly. Don't just leave it at less glucose is produced, finish it off, so less proteins are made. So remember, in plants, you don't need this bit, if I just explain it, in plants, glucose that's made can be converted into amino acids, and then those amino acids are then in turn converted into protein. And we know protein is used for growth and repair. Okay, question 5 is looking at the heart. So this is your organization, circulatory system. So which blood vessel transports blood with the highest oxygen concentration into the heart? So let's just add arrows on. So remember, the right atrium receives blood. Blood enters the valve and then out into the lungs. Here, blood enters from the lungs. Okay, so that's out. Here, blood comes from the lungs, and then it leaves via this, which is called the aorta, to the body. Always best to add some arrows on. So let me just neaten that up a bit so you can see the arrowheads. So that's in, out, in, out. Always remember, if I've put a star, that's your atria or atrium, and atria always receive blood, and ventricles, which is these chambers, they always pump blood out. So the question is saying, which blood vessel transports blood with the highest oxygen concentration into the heart? Well, it has to be D, because the blood is coming from the lungs. So this blood here is high in oxygen. Blood pressure is a measure of the force of the blood against the walls of the blood vessels. Which blood vessel transports blood at the highest pressure? It must be the blood vessel that takes blood away from the heart. So let's have a look. Away from the heart is this one, C, which is your aorta. What is the correct order for blood flowing through the heart to the lungs? So we're looking at this route. So we are on the right side. So when you're looking at the heart, okay, it's your opposite. Okay, so that's my right, that's my left. So I've got my right atrium, I've got my right ventricle, and then it's going to the lungs, so that must be my pulmonary artery, because it says through the heart to the lungs. So let's have a look at the right. So we know it can't be that one, can't be that one. Right atrium, right ventricle, and it should be pulmonary artery, so it's the third one. Question 5.4. Every year, thousands of people in the UK have heart attacks. A heart attack is caused when the heart muscle cells do not get enough oxygen. Statins and stents are two treatments to reduce the risk of someone having a heart attack. Okay, so statin is a tablet, and stent, that involves a surgical procedure. You need to know the difference between the two. Evaluate the risk of statins compared with the use of a stent to reduce the risk of a heart attack. Evaluate question, you need to have advantages, you need to have disadvantages, and then you need to have a justified conclusion at the end. So let's start off by looking at advantages of statins. So like I said, statin is a tablet, so you can say, oh, it's easy to take. And it's a tablet, which means it's low cost, it's not going to be expensive. And you can also mention the role of the statin. It will decrease blood cholesterol, because you need to know the purpose of statins. So remember, it's this cholesterol that builds up inside the blood vessels, so coronary arteries, which then leads to a blockage of the blood vessel, which reduces or stops the blood flow to the heart muscle, which then leads to the heart stop working, which is a heart attack. Okay, so that's advantages. Let's now do disadvantages. You are allowed to draw out a table, which really I should have done at the beginning, but it's fine, we'll just do bullet points and subheadings. Disadvantages of statins. You might forget to take a tablet, so you have to take this tablet every day. So might forget to take the drug. And you can talk about side effects of drug. And that should be enough for statins. Let's do the same for stents. So advantages of stent. Okay, so with this one, you can talk about the effect of having a stent is immediate. So you see results immediately. Involves a rapid recovery after the operation. And what's the purpose of a stent? It holds the blocked artery open. So you can mention that. Holds the blocked artery open, keeps the blocked artery open. And then disadvantages. So it's a surgical procedure, so you can say anything in biology which involves an operation, always write about risk of infection from the operation and blood clot, risk of blood clot. And again, that's with any surgical procedure where a person would be left immobile for a long period of time, basically when they can't move. Okay, risk of blood clot. And that should be it. Okay, and finally you can say, I conclude, there's no wrong or right answer for this part, so I would say I conclude that statins are a better option because they don't cost much. So what you're doing is you're repeating some of the things that you've already said. So they don't cost much and are easy to take. Just a bit of repetition, but you've already made the point, so just rewrite it out again, and that's it. Okay, nice, easy, straightforward. Many people who survive a heart attack get out of breath easily when they exercise gently. Explain why heart attack survivors get out of breath easily. So here, so talk about the heart muscle cannot contract because it's not as strong. So heart muscle cannot contract. What does that lead to? Less blood pumped out of the heart. So there's less blood being pumped out. That means there's less oxygen for what? Respiration. And the question does talk about, so take it back to breathing. So breathing rate increases to supply more oxygen. Done. Question 5.6. Scientists have developed patches of beating heart cells to repair damaged heart tissue. The patches are placed onto areas of the heart where cells have died. New cells grow to replace the dead cells. The patches are made using a person's own cells that are converted into stem cells. Explain why stem cells are used to make the patches. So this question is looking at the properties of stem cells. So what do you know about stem cells? So stem cells are undifferentiated cells. Stem cells are undifferentiated cells. Second mark. So they can form heart cells. That's what stem cells can do. Stem cells have the ability to convert themselves into any cell of the body. So the scientists could have used human embryonic stem cells to make the patches. Give 2 advantages of using stem cells made from the person's own cells rather than using embryonic stem cells. Well, cells will not be rejected. And here is, because we're not using embryonic stem cells, there's no risk of damage to embryos, or you can say there's no human life destroyed, or no potential human life destroyed. Right, question 6 is about plant transport systems. So describe how water is transported from the soil to the atmosphere through a plant. Okay, so if you just have a quick look at a plant. So the question is saying, how is water moving up and then leaving the plant? So basically this is looking at the transpiration stream. You're just describing it. So you need to say that water is absorbed via root hair cells by osmosis. Yeah. How does it move through the plant? Travels through the xylem. Xylem carries water and minerals. Travels through the xylem and then continues until it gets to the leaves, and then obviously escapes via stomata, lost through stomata. So that's my 3rd bullet point. And this is called transpiration. This is transpiration. And that should get you 4 marks. So the movement of water through the xylem is called transpiration. What do we call movement of dissolved sugars through the phloem tissue? Translocation. Phloem tissue is made of sieve tube cells and companion cells. There's your diagram. Okay, companion cells have lots of mitochondria. Explain one way sieve tube cells are specialized for their function. This links to cell biology as well, where you looked at specialized cells. So let's have a look at the sieve tube cell. So look here, you can see pores. So they have pores. And this part here is called the end walls. So have pores in the end walls. So it's 2 marks. So sugar, so dissolved sugar can move from cell to cell. Because this way, there's a reason why they've given you a diagram, so use that diagram to answer the question. What does the structure of the companion cell suggest about the process that moves dissolved sugars through the phloem tissue? Give a reason for your answer. There's lots of mitochondria. Mitochondria means that, so if a cell has a lot of mitochondria, it means that there's aerobic respiration happening a lot in this particular cell, okay, which means that it's probably using active transport. So it's 2 marks, so you need to say this is suggesting that this process requires energy, or the cell carries out active transport because the cells have lots of mitochondria. Describe why it is important that dissolved sugars are moved both upwards and downwards in a plant. So here you need to say that sugars or glucose is made in the leaves, which are found mainly towards the top of the plant. So sugar is made in the leaves by photosynthesis, and all cells need sugar for respiration. And your 3rd mark is, you can say sugar is transported, because sugar is needed by every single cell, so it needs to move around the entire plant. So sugar is transported for storage as starch, fat, or oil. We can just write 1, that's fine. Okay, or you can say sugar is transported to meristems for growth, or for cell division, or for mitosis, but this is an easier bullet point to jot down. And that brings us to the end of the paper. Okay, so just to give you an idea, so this is a 2021 paper. Okay, so if you had a go at the paper, just to give you an idea of the grades, so this is June 2021 paper. If I take you through the grade boundaries for 2023, so 47 and above, so this paper's out of 70, so 47 should get you a grade 9, 40 to 46 gets you a grade 8, 34 to 39 gets you a 7. 28 to 33 gets you a 6. 22 to 27 gets you a 5. And then 16 to 21 gets you a grade 4. So it's out of 70. If you hit half the marks, roughly, which is 35, you would get a low 7, a high 6. Okay, remember grade boundaries change every year. There are no grade boundaries for 2024. Grade boundaries are generated once everyone sits their exam. But what I've shared with you here is readily available if you just Google it. Okay, these are grade boundaries for 2023, just so that you have a kind of a rough idea of what grades you are getting when you are trialing these papers. Okay, thank you for watching. I shall be sharing more papers over the next 2 days. That way you get a greater variety or greater exposure to the different biology papers that are out there. So thank you for your time. Thank you, everyone.